Hello everybody, I wish everything's fine. Today we'll talk about very important topic in bioinformatics. We'll talk about multiple sequence alignment. Multiple sequence alignment, or MSA, is one of the very important topics in bioinformatics. It's very useful for biologists and bioinformaticians, and you can get a lot of result out of it and very useful for interpreting many information in bioinformatics and biology. So there is many things we will talk about during the coming lectures. First is how to gather sequences, what is the difference between different programs, multiple sequence alignment programs like Cobalt, ClustalW, TCAFE, Muscle, MAFFT, and also how to create and compare multiple sequences that, of course, cannot be aligned. That's also one of the things that is very important. Also, in the outcomes, we will learn why multiple sequence alignment is useful for scientists. Also, how to identify situations that multiple sequence alignment don't help. The main criteria for building multiple sequence alignment, what is the main application of multiple sequence alignment? What are kind of sequences that we are looking for? Of course, this is very important. Tips for naming the sequences, how to name sequences in the right way. Also, tips for difficult multiple sequence alignment to interpret, how to interpret the multiple sequence alignment, and the difficult multiple sequence alignment, how to interpret it in the right way. Also, one of the things what we would like to learn during the course of this module is comparing sequences you cannot align, the sequences that cannot be aligned, how to interpret this and how to compare this kind of sequences. Okay, now we will how to build multiple sequence alignment. This is not the first topic. Of course, building a multiple sequence alignment is like the Swiss knife for MacGyver. MacGyver is a very famous series in the 90s, and during this series, the hero in this series usually build many important things and useful things for his missions using Swiss knife. So he have a very simple tool, which is a Swiss knife, and this Swiss knife, in all episodes, he uses Swiss knife during his adventures for making very useful things for his missions. So the same thing we think about multiple sequence alignment for bioinformatician. It's very useful for bioinformatician in many different situations to help bioinformatician in making their life of research easier for them. Because it simply can facilitate many important applications. Okay, building multiple sequence alignment, as we say, uh, always it's a far from exact science. It's art. So it's more art than science because simply it's requiring use of everything you know in bioinformatics, also in biology. So it needs a very good information in biology and bioinformatics. Now we will identify situation that multiple sequence alignment doesn't work. It doesn't work first if you are trying to assemble in assembling sequences pieces for sequencing project, multiple sequence alignment will not be useful in this situation. Also, to turn EST, cluster, expression sequence tag, cluster into a gene sequence. In this situation, multiple sequence alignment also is not useful. When sequence, if you're interested in, has no homologs, also in this case, this multiple sequence alignment cannot be useful. So in this case, we will find something else. We will tell you about it, such as, for example, we will look at the functional criteria and conducting what we call pattern search. And this will come to this later on. Okay, now we'll see the main 5 steps for building informative alignment. Number 1 is gathering your sequence. Number 2 is compute multiple sequence alignment. Number 3, evaluate the quality of your alignment. Number 4 is interpreting your multiple sequence alignment. Number 5 is keep the sequence for further analysis. And we'll talk about most of these items and most of these steps later during the lecture. Okay, now we'll talk about very important thing now, which is what are we looking for with multiple sequence alignment? Of course, the main idea of multiple sequence alignment to put amino acids or nucleotides in the same column because if they have similar position according to some criterion, they will have similar function. So for that, we'll tell you about the four major criteria to build a multiple sequence alignment that all have different properties. Okay, first, which is same structure similarity. That's what we are looking for in the beginning. This is the first thing we are looking for. If amino acid play the same role in the structure, they should be at the same column, at the same position. What we call in the structure superposition program, that these programs are always looking for this criteria to try to find similarity between sequences of multiple sequence alignment, just to try to prove that there is a structural similarity. So, for example, if we have positions of certain amino acid in a certain position and across many species or many sequences, this will give us the idea that maybe these sequences play a structure function, or what we call structure rule for this protein or for this DNA. So this is what we're looking for as what we call structure similarity. This is the first thing we are looking for. Number two is what we call evolutionary similarity. Also, if you have amino acid or nucleotide also the same columns, this in this common ancestor of all sequences, this in the same column, this also can give us indication that there is evolutionary similarity. And also we have other programs that respect this criteria. The number three is functional similarity. Also, we have, if we see that amino acid or nucleotide have also the certain position and have very similar structure and at the same position, this also gives us a criteria that there is a functional similarity. If these amino acid have, of course, indicate some function or associated with some function, and there is many programs that can respect this respect this criteria. Number four is sequence similarity. This is the fourth criteria, which is very important. That if, of course, most of the program is looking for this criteria, which is we are looking for amino acids or nucleotide that in the same column. This criteria is the easiest criteria that we could look for using most of the multiple sequence alignment program. Now when the sequence are closely related, we will see, of course, that there is structure and evolutionary and functional similarity. And all these three, the structure, evolutionary, and functional similarity, are equivalent to the sequence similarity. Because if there is a sequence similarity, you can see and you can be certain that there is structural and evolutionary and functional similarity. So the key and the most important thing in this four criterion is number four, which is the sequence similarity. Now, there is many application. We'll talk about it just briefly. And main application for multiple sequence alignment. Number one is extrapolation. This application means that a good multiple sequence alignment can help to convince you that uncharacterized sequence is a really member of a protein family. So if you have, for example, a sequence and you would like to know if this sequence belongs to a protein family, simply you just use some software like X++ or something that will let you know which if this multiple sequence alignment, you're using one of the uncharacterized sequence, is a part of a family and have most informative domains that belong to that family. If I have uncharacterized sequence, it simply can tell you if it belongs to other family or certain family in the database. Also, one of the things that is very useful is the phylogenetic analysis. Phylogenetic analysis means that the carefully choose sequences you could include in your analysis. Multiple sequence alignment can reconstruct history of this protein of, for example, I mean, by using multiple sequence alignment, you can simply build a phylogenetic tree, and with this phylogenetic tree, you can indicate which protein or which sequence is most related to your sequence. So if you have a sequence, either DNA or protein, and other sequences you would like to compare with, you can simply see the phylogenetic relation between your sequence and the other sequence. So that's the meaning of that's one of the very good important application for multiple sequence alignment. Also, one of the good application is pattern identification. This by discovering very conserved position, of course. By this, you can identify the region that is characteristic, for example, characteristic of function of that protein or even amino acids. For protein or nucleic acids. So if you have a genes or if you have a proteins and you would like to know what is the pattern that exists through all these proteins, group of proteins or group of genes, you can simply use multiple sequence alignment. Some programs like WebLogo, we'll talk about it later. Simply, you can gather your multiple sequence alignment, put it in a program like this, and it simply can detect what is the pattern for your sequences. Also, one of the very important thing and very similar is domain identification. Domain identification, uh, also some programs like ProSite. You can gather your multiple sequence alignment and you can identify pattern or domains, and these domains are like can be identified easily through multiple sequence alignment, and these domains is characteristic for your sequences. You can use some programs like ProSite, as you can see here. Also, you can find if you are using protein, if you are using DNA, you can find DNA regulatory elements by using the DNA sequence. If you are doing multiple sequence alignment for some genes or DNA, for example, and you are just try to identify uh, uh, some position for the regulatory element. This also can be easily found using some program like Gibbs Motif Sampler, this very important program, and can easily identify these positions. Also, you can use it for structure prediction. Well, this is one of the very useful application. It's a very good and very useful. You can predict what is the position for if you are comparing, for example, a multiple sequence alignment for amino acid sequences, different proteins. If you would like to see what is the conserved region for the beta pleated sheet, alpha helix, stuff like this, this also can be useful in structure prediction using multiple sequence alignment. Also, NS-SNP analysis, like synonymous and non-synonymous mutation, also can be very useful. You can see the synonymous mutation or non-synonymous mutation using multiple sequence alignment if you're comparing different genes, for example, and would like to see what is the position for NS-SNP analysis. Of course, we have some program like SIFT and other programs that can be used for this application. For PCR analysis, multiple sequence alignment can be used to identify less degenerate portion of the protein family. So in this case, you will find where is the place that we can design useful primers and to isolate certain genes or certain positions or certain location for DNA. So it's very simply we can do multiple sequence alignment to identify this portion for a protein family. And of course, Good Hope, that's one of the important programs that can be used for this purpose. Also, you can see you can you can understand that there is many sequences we are looking for when we are making multiple sequence alignment. It's very important to use amino acid, uh, a protein sequence, if possible, always. And to import amino acid or nucleotide are not allowed to mutate. I mean, we're always looking for positions of protein that there is no mutation, so there is no differences. And this position, which is conserved, usually it's the most important positions in your protein. So that's the position that we are looking for. We are always looking for the position that are conserved across multiple species. Because sometimes like there could be active sites for proteins, for example, and less important position, this always the position that is easily changes. So when you are looking for a multiple sequence alignment, always try to focus on the position that are less degenerate. I mean, so which is mostly conserved. So do we have some tips for naming sequences before using multiple sequence alignment program? Try to be sure that your sequences names never use white spaces, don't use special symbols, never use names that longer than 15 characters, never give the same name for two different sequences. This is very important because these four tips are very important. If you make any mistake, you will have a problem in using any program for multiple sequence alignment. So try to stick for this with these tips, and so to be sure that you will not have problem using any program for multiple sequence alignment. Also, for difficult multiple sequence alignment to interpret, you have three very important also tips. One of them is remove insertion or deletion, and we will tell you how because it's very important to understand where and how to do it. Redo multiple sequence alignment for smaller set. If you have a smaller set and you would like to know which one of your sequences causing trouble in multiple sequence alignment, you can have a smaller set, and also keep trimming to interpret, especially for the extremities. So always to enhance your multiple sequence alignment, you can remove gaps, remove extremities, and keep informative blocks, as we said, because it may give you a very important indications for conserved region, which we'll talk about later in the coming lecture. Now, this is, you can try in your own time how to search for sequences. Simply, I give you this example. You can go to UniProt and the link is available here. You can just go and type heat protein 70, HSP 70, and you will have here, as you can see, you can gather your sequence here. You will have here many of them. You just try to pick the sequences you would like to use for your research to study this protein, and then try to export it to as faster format. And of course, you can use a different program like ClustalW or TCAFE or MAFFT. We'll talk about them later, and try to generate a multiple sequence alignment using the sequences that you just extracted from the UniProt database. You can do this in your own time. Also, you can select protein 70 from human, rat, mouse, Arabidopsis, chicken, pig. So they are different species. These different species, but you have, for example, the heat shock factor 1 from these different species in FASTA format, of course, and use these sequences to build a multiple sequence alignment. And of course, you would like to know what is or where and what are the important positions within these sequences, and you can identify it simply by multiple sequence alignment. What is the conserved domains and conserved positions within this sequence. Now we will have something else. This links is like a flashcard and quiz set for bioinformatics lectures. Of course, these four links that's generated by AI. The first one is how to, of course, how to interpret for this. These four links will be with us through all the coming lectures, so you can use it just to study and to practice. It give you a lot of questions that you can answer and test yourself and see the right answer using some important like building queries and building quizzes using very important AI tool. I hope you enjoyed. You can just uh, open it and practice with yourself in your own time. Thank you for listening for this lecture, and we'll continue with other lecture now. I hope you enjoy the lecture and try to solve some of the questions. Thank you, and see you in the coming lecture.